Hey guys, welcome back to session 2 of Living Environment Regents Review, sponsored by Learn America. Today we're going to complete our session of Journey Through the Cell. We're going to start with our journey through the nucleus. From the nucleus, we're going to look at how genes and traits are inherited, and then from there we'll take a look at how the body systems are created. So right now, if you were here with us the last session, we're going to start in the packet number 1 on page 11. So you could go back to the first one. So as you guys could see, once again we're going to travel through the nucleus, review DNA, protein synthesis, transcription, translation, any changes that may take place, such as mutation. We're going to visit the centriole and talk about the importance of the centriole in the cell, in its role in reproduction. Then we're going to take a look at all the body systems. Okay? All right, good. All right, so last time we took a journey through the cell. We saw the mitochondria, the chloroplast. We traveled across the cell membrane. In the cytoplasm, we took a look at all the different biochemical processes that takes place. So right now, we're at the nucleus. And so if you take a look, here's the nucleus singled out by itself. You have the nuclear envelope, nuclear pore for materials to pass in and out of the nucleus. We have the nucleolus. Chromosome is also found inside the nucleus. And we're going to find strands of chromatid inside the nucleus. We're going to find out that the nucleus is there, will affect the type of genes and traits that's created in the cell. So we're going to see it will determine what proteins will be made in the cell. We're going to visit the purpose of the protein, how protein will serve as a source of enzyme. And we're going to also see that it will determine what type of chemical reaction will occur inside the cell. So let's continue with the nucleus. Here's a quick review of genes and traits that's found inside the nucleus.
Each chromosome contains many genes. Each gene on the chromosome contains both exons and introns. The exons contain the coding sequences, and the introns contain the non-coding sequences.
So as you're looking inside of the nucleus, you'll find the chromosome. So when you're preparing for the state test, it's important to remember the structure of the chromosome, be able to identify it, be able to identify what it's made up of. We're going to see that the chromosome is actually coiled up DNA strand. So it's the DNA strand coiled around protein, condensed into a chromosome strand. If you unwind the chromosome into this long strand of DNA, different sections on it code for different genes and different traits, your characteristics. For example, the type of enzymes your body could make, the color of your hair, the color of your eyes, all of those characteristics is based off different segments of the DNA. So let's go back and review parts of the DNA. So the DNA stands for deoxyribonucleic acid. We're going to see that the DNA is our hereditary material. That's what's passed from one generation to the next. It's the instructions on how to make the different proteins in our body. So here's another look at the chromosome itself. And here we could see that the chromatid, you have one side represents a chromatid. Here's the other chromatid. Together they're referred to as sister chromatids. Holding it together are the centromere. So centromere holds the parts of the chromosome. So feel free to take notes along the page, write down things that you may not have remembered throughout the school year. All right, so let's take a look at the DNA structure. The DNA, once again, it's a double helix. There's 2 sides to it, 2 opposing sides, and we see that it's made up of bases, nitrogenous bases. Here's a closer look at that structure. The DNA, the building block of a DNA is a nucleotide, a DNA nucleotide. The nucleotide, this is a structure. So you should be able to identify this structure. This is on page 11 So some of the structures you're going to add in yourself, just so that you're following along. So I'm at the bottom of page 11 in the old packet, in last section packet. So here's the nucleotide. The nucleotide, you need to be able to identify the structure as is. So be able to identify what structure A, B, and C. Do you remember what these are?
One of them's glucose, right?
All right, so one of them, it will represent the sugar inside. So here, come on in. So you'll have the sugar. This is the 5-carbon sugar. And so for DNA, that 5-carbon sugar represents deoxyribose. Attached to it will be the phosphate group, and attached on the other side, represented by C, are the nitrogenous bases. We're picking up from page 11 from the last time. Then we'll say that again.
That part's on 12.
Is on 12. Okay, the image is on 12. So the nitrogenous bases for DNA, there are 4 types. You have adenine, thymine, guanine, and cytosine. And so you would find one of the nucleotides on one side of the DNA ladder, and then you would find the opposite, the complementary side to have the opposing setup of this nucleotide. For example, if in this nucleotide we had adenine, on the complementary side we would have thymine. They're bound together by a weak hydrogen bond. So if it was guanine on this side, it will pair up with cytosine. And so we usually use the letters to represent it. So A pairs up with T, T with A, G with C, C with G. And this is for the DNA strand. Now, when it's time to make a new copy of the DNA, say you produced a new cell, you need to make a copy of the DNA for the new cell. So in order for that to take place, DNA replication has to take place. And that's where the DNA strand will then split by the weak hydrogen bond. And so we refer to that as unzipping of the DNA. So you'll see the DNA unzip, the new strand will form using the base pair of A to T, G to C, and so forth. So let's take a quick look at a review of
DNA replication is a fundamental process by which multiple copies of DNA are formed. The double-stranded DNA is separated by the enzyme helicase. Single-stranded binding proteins keep the strands separated. In the leading strand, there is a continuous synthesis of the new strand, while in the lagging strand, there is a discontinuous synthesis in the form of Okazaki fragments. RNA primases create an RNA primers, and DNA polymerase III facilitates the synthesis of the new DNA strand. The DNA polymerase I replaces the RNA primers with DNA. Finally, the Okazaki fragments are linked together by the enzyme DNA ligase.
So your takeaway from that should be that in order for the DNA to split, an enzyme is required. DNA polymerase is the enzyme that's required to split the DNA. So the basic that you'll need for the living environment exam is to understand that that process is controlled by an enzyme, right? And it splits by the weak hydrogen bonds. Let's do a quick practice question on this. We have here, hereditary information is stored in the genes. Genes are made up of blank. This blank can be replicated or control the production of blank. So this is a review question in your packet on page 12. So let me give you guys a minute to look that over. So your question will be, which molecules are represented by box A? What type of molecule in box A serve as a template? Explain what this means. And which molecules are represented by box B? Take a second, think about it.
hour in to just take a break?
Yeah, if you just give me the... All set? What do you think? What molecules are represented by box A?
DNA.
Yes. And so that statement, a type of molecule in box A served as a template. What does it mean that it served as a template?
It shows them how to copy it.
Right. It's going to serve as the copying source, a source for the new strand to be copied from. And which molecule is represented in B? What do you guys think about this one? No? Thinking about it? So let's see. So if that's DNA, DNA can be replicated and it controls the production of where we started.
Cells.
No, it doesn't produce a cell. It has the information for that cell. So B represents protein, is an example of something that would be made from it. Okay. All right, let's do another practice question, talking about base pairs. If 15% of the DNA sample is made up of thymine, what percentage of the sample is made up of cytosine? I'll give you guys a second. There's no marker. You should have an expo marker over there. Thank you. Yep. Did you come up with an answer?
Yeah.
What do you? 50. What do you think?
Yeah.
Same?
Yeah, same.
All right. So let's take a look. So it says 15% of the DNA is made up of thymine. So if 15% is T, what percentage would be A? Think about the base pair rule. T always pairs up with A. So if there's 15% T, there's going to be also 15% A. You guys see that? Because A and T, T and A are base pairs. What percentage is left over? Because there's 15 and 15, so that's 30. So you have 70%. Right, so 70% is left over. That 70% can be made up of G or C. We want to know how much is made up of C. So divide that in half, and you get your 35% would equal to C, which gives you the other 35% would be G. So the correct answer, what percentage of the sample would be made up of C? 35%, right? So this is an example of understanding base pair. Whatever the percentage is for T or A, the complementary base would be the same amount. And so from the remainder of that, you could determine the rest of the bases. Yeah? All right, so make any corrections if need be. All right, so let's move on to the ribosome, because we'll talk some more about genetics later. So we're going to travel from the nucleus and go over to the ribosome. The picture we're used to seeing, here we are, the ribosome. It has a large subunit and a small subunit. So the ribosome is where protein is made. It's the site of protein synthesis. And let's take a look at the process. So here I wanted to give you a glance as to how the process will take place. So here the nucleus, DNA has to send the information over to the ribosome, to instruct it on how to make proteins. So this is accomplished through a series of steps. The DNA will get transcribed into messenger RNA. The DNA cannot leave the nucleus. There are other enzymes that will break down or destroy the DNA, so it cannot leave the nucleus. So in order to get the message there, a messenger will come into play. So the DNA gets transcribed into a messenger RNA. The messenger RNA is able to leave the nucleus, travel into the cytoplasm, goes over to the ribosome where it will get translated. And what's translated? The code is translated into amino acids. The amino acids are then combined to form proteins. So this is a quick glance to a complicated process. The introduction of the messenger RNA comes into play here. RNA helps to transfer the information that's needed for protein synthesis. There's 3 types. There's the messenger RNA that's going to take the message from the nucleus over to the ribosome. There's a transfer RNA that's going to help to transfer the code to produce the amino acid. And you'll find the ribosomal RNA inside the ribosome itself. For our purpose, for the state exam, the 2 that's talked about the most and discussed the most is the messenger RNA the most, followed by transfer RNA. But predominantly, most of the questions are based on the messenger RNA. Here we go. So the RNA, similar to that of the DNA, so we're on page 13, similar to that of the DNA, it's made up of a nucleotide. So it's made up of an RNA nucleotide. The structure looks the same, right? Can you see that? What's different about it? About the makeup? Between this and DNA? Right? Same as DNA? Right. So we have a change in the bases and we have a change in the sugar. So we have the phosphate group represented here. Ribose instead of deoxyribose is found in the RNA. And we have the nitrogenous bases, which are here. So the nitrogenous bases, instead of thymine, you're going to find that we have uracil. All the other bases remain the same. Here's the packet. All the other bases remain the same, so there's still adenine, guanine, and cytosine, but thymine is replaced by uracil. Uracil is replaced in thymine, so what we'll see for base pairing is that A would now pair up with U instead of T. G would then pair up with C. So let's take a look at some of the differences. I'll give you guys a second. So you could see DNA, there's the double strand. Whereas the RNA, it's a single strand. DNA contains thymine, RNA uracil. DNA, A is paired with T, whereas in RNA, A with U. And there's a difference in the sugar. Deoxyribose in DNA, ribose in RNA. So let's take a look at the transcription process. So transcription is the copying of the genetic message from a DNA into a messenger RNA code. So let's do a quick practice with that. So if the DNA, let's say, is ACC, what would your messenger RNA be? UGG. Or let's say our DNA is TA. What do you? No, go ahead. Go ahead. You. So here, a lot of people think that, but it can have A and Right. We just can't have any T's in the RNA. So T still pairs up with A, but A pairs up with U. So this process is called transcription. Transcribing the message from DNA into RNA. The RNA now will travel out of the nucleus to the ribosome, where it will be translated. So translation, you're going to use the messenger RNA molecule to build a protein molecule. And so once it gets there, you read every codon, every 3 base will make up a codon. And that's what you use to determine the amino acid sequence. So let's do a few practice. So here my message, I'm going to move it over here. My messenger RNA is, the first one is U, G, G. My second one is A, U. Every 3 represents a codon. So that's the code that you'll use to translate the message. So use your sheet and let's look up what amino acid is it translated into. UGG. So there are different scales that you can, different charts. This one you'll go from the first letter, second letter, third letter. So the first letter, here's U. What's the second letter? G. It's up here. And the third one? G. So it's going to bring us in here. U, G, G. And here we go. Thyrophane. So what you need to do is you only need to write the first 3 letters for our purpose. The amino acid will be attached to the transfer RNA. The transfer RNA will bring the specific amino acid into the ribosome. And we'll take a look at that in a second. Let's try another one. Let me write my transfer RNA. What amino acid will it bring? AAU. You see it? Asparagine, here we go. ASP. So in the exam, you will be provided with a chart for you to use. You don't have to memorize any of these. You just need to remember, focus on the messenger RNA to get the amino acid. You don't use the transfer RNA. Only use the messenger RNA. Think about it. To get the message, you use the messenger RNA. So decode the messenger RNA for the amino acid. Any questions on this? Okay. Let's take a detailed look at the process.
The stages of translation are divided into initiation, elongation, and termination. Eukaryotic mRNA has a unique 3-end poly A tail and a methylated cap at the 5-end. In the initiation step, the mRNA binds with the small ribosomal subunit. The second step is elongation, which requires the presence of tRNAs. tRNA molecule contains an anticodon, which is complementary to the codon found on the mRNA. The mRNA start codon is AUG, which corresponds to the amino acid methionine, which is attached to the appropriate tRNA.
So those are the amino acid.
The large subunit of the ribosome now binds with the smaller subunit to create the peptidyl or P site and the amino acyl or A site.
So here's your messenger RNA, tRNA, those are the amino acids.
tRNA occupies the P site and the second tRNA occupies the A site. Methionine is joined to the amino acid that is attached to the tRNA.
So all the amino acids
the free tRNA
attach.
occupying the P site are now exit. As the ribosome moves along the mRNA, each amino acid attaches to the next amino acid in the A site. The elongation step continues until the tRNA encounters the stop codon and then a releasing factor binds to the A site. Translation is now terminated. During termination, the ribosome dissociates and the newly formed protein is released.
All right, so that was a good look. What we didn't do in the last section was combine the amino acids we formed. What was it? It was I thought it was tyrosine and asparagine. Those were the two. They would combine to form a protein. You combine the amino acids that's made using a peptide bond, and together they make a chain, protein chain. And so here, that's what's being shown in this image here, where the protein chain is made. Protein, based on is determined by its shape. The shape will also determine its function.
The stages of translation are divided into initiation, elongation,
And we saw it. All right. So if there's any change in that sequence or that message, it's referred to as mutation. Any change in the code. And let's take a look at that. For example, here's our messenger RNA strand. And so you determine the amino acid based on every 3 codon. And let's take a look where we have, here's the code. CAA codes for this particular amino acid. There was a base change. Instead of it being CAA, the C got changed to an A. And so that changed the type of amino acid that's going to be made. And that change will change the function and the structure. Sometimes it won't cause a complete change in the protein itself, but most of the time the mutated sequence will. Here's another type. We could see where the C is changed into a G, and that creates a completely different amino acid. Insertion is another type of mutation where a letter can be inserted. So if we take a look up top, we had CAT, coding for histone. Here we're going to insert A. So since adenine was inserted in, it changes the sequence. So here the first 3 are the same, but instead of it being CAT, now it's going to be ACA, which now codes for a different amino acid, which changes the sequence of the amino acid, changes the type of protein that's going to be made. Deletion is another type of mutation where, let's here, take a look, CAT, which codes for histone. The C gets deleted out, so that leaves you with ATC, which is the code for stop. So as translation is taking place, there will be a code for stop, telling your body when to stop producing that protein. For example, when you have milk, your body will make lactase, the enzyme lactase, to break down dairy products. Anyone lactose intolerant? No? All right, so if you're not, your body naturally makes lactase. Now, do we need lactase all the time to be active? That's not true. So when your body's ready to make that enzyme, it will go through the process of translating, producing that enzyme. But when it's not required anymore, there is a code to tell your body to stop producing it. All right, so let's do a couple practice questions. First one, and this is going to take us to page 14 in our review packet. Building block 1, 2, and 3, or A, B, and C, forms protein. Identify the type of building block present in A, B, and C. Second, if the sequence of building blocks are changed, what effect could it most likely have on the protein? So go ahead, take a, yeah, it's the one from last time. I'm finishing up. Do you need a copy of it? From last week?
No, this one.
Yeah, it's from last week. Yeah, yeah, I have a copy here if you need it. Let's pass it back. Yep. Yeah, we're finishing up that section. So go ahead, page 15. Building blocks for protein. All right, so what do we need to make proteins? What would the building blocks be? What do you guys think? This is one of the popular questions or common question to see. What things would join together to form protein? Amino acid is right. So amino acids join together to form protein. What would happen if there was a change in one of the amino acid? How would it affect the protein? You will see that it would change the function, the shape would be different because the amino acid was different. Now let's do another one. What's the relationship between the three? We have a cell, DNA, and protein. Take a minute, think about this. All right, what do you think? What's the connection? Oops. You see it? Right, so if we think here's the cell, where's the DNA found? Inside the cell? Right, and the DNA codes for what? Right, so it codes for the protein. So the DNA is found in the cell, the DNA codes for the particular specific protein that will be made. So that's the connection between all three. All right, so we're going to leave the ribosome and we're going to head over to the centriole. So the centriole now is involved in cell replication. It's time to make copies of the cell. The centrioles are these rod-like structures that's found in the cell, and they're responsible for making spindle fibers when the cell is ready to replicate. And those two processes, the first one we're going to look at is mitosis, also referred to as mitotic cell division. Mitosis is when the cell replicates, create an identical copy of itself. So you'll end up with two identical copies. And based on the process, the cell, in order for the cell to do that, it will go through process of mitosis. And so for mitosis to take place, there are different phases. There's interphase, which is known as the resting phase. During interphase is where we have a growth phase. The DNA replicates, and then it prepares for division. The active portion, the active part of mitosis is prophase, metaphase, anaphase, and telophase. So I haven't seen questions on the phases themselves in years, but you need to understand what's happening overall in the process. What's the result of the process? So let's take a minute and look it over.
Mitosis is the process in which a eukaryotic cell divides into two cells. The two daughter cells contain exactly the same number of chromosomes as the parent cell. During new cell formation, the period of cell growth is called interphase. During this phase, the DNA is replicated. Prophase is the initial stage of mitosis. During prophase, the nucleolus disappears and the chromatin condenses to form chromosomes. The centrosomes start to move towards the poles, organizing the spindle microtubules between them. By the end of prophase, the nuclear membrane breaks down and disappears.
So we could see where the centriole here and here will attach.
Attach to the centromeres of the chromosomes, and the chromosomes line up along the metaphase plate. Anaphase starts with the contraction of spindle microtubules, which split the chromosomes into sister chromatids.
This guarantees equal amount of information on each side.
Start to move towards the poles. During telophase, the chromosomes reach the poles and the nuclear membrane is reformed. Cytokinesis begins with the formation of a cell plate. Following cytokinesis, two daughter cells are formed that are exact copies of the parent cell.
Okay, so the main thing was to see the centrioles in action, where their job is to help pull those chromosomes apart. Remember, you're not going to be asked exactly what's going on in each phase, at least for this, the exam, the living environment exam the way it is right now. You just need to understand that through this process, you will end up with two daughter cells. These two daughter cells will have the exact number of chromosomes. They will be identical to each other. So think about the outcome or the purpose of the process. And so that brings us into asexual reproduction. There's certain living things that reproduce using the process of mitosis. And so as a result, these the offspring from the parent cell the offsprings are identical to the parent cell. It involves only one parent. So imagine that one cell will split, go through interphase, prophase, metaphase, anaphase, telophase. At the end, you end up with two. So there's certain organisms that go through that phase, and that's how they reproduce. This is not involved when you're producing gametes like the sperm and egg cell. The offsprings are going to be identical to the parent. Some advantages to asexual reproduction is that the single individual may produce rapidly. So we see bacteria falls into this category. The disadvantage is that there isn't any genetic variation in asexual reproduction, little to no genetic variation. And so some examples of that, I gave it to you in your packet. So right now we've moved on to packet 2 from today. Examples of this, we have regeneration, we have binary fission, sporulation. Runner is another example of asexual reproduction. But the main thing I want you to walk away from this is that in asexual reproduction, little to no variation, rapid reproduction takes place here, and the cells are identical to their parent. So let's take a look at this. Here's your practice Regents question. Here's the organism, here are the two offspring. In the circle representing organism 1 and organism 2, write the number of chromosomes that result from the normal asexual reproduction of organism A. So what would be this? I want you to write it down. You have this chart on page 1 of the new packet. Go ahead and write it in.
Yeah, Ms. Johnson.
Yep.
Binary fission.
But it's listed on the first page.
Okay.
It's in the last. Yep, there you go.
All right, underlining.
Yeah, and usually you will see that in a reading passage on the exam or leading up to a question like this.
All right.
So what did you guys put in each circle?
I put this.
You chose 8? What'd you put?
I have 4. I said 8.
All right, why'd you go for 8?
Because they're going to be identical.
All right, and so that's the point. Because, so you're going to look for, the words that you're going to look for, asexual reproduction, key words to think about, mitosis. When you see those, you automatically think identical. It's going to look the same, even if they gave you a triangle in the parent cell. The offspring will be a triangle. It has to be identical. Let's go on to sexual reproduction. So sexual reproduction, on the other hand, involves 2 parents. It now involves the union of the gametes. This is the sperm and the egg. The offsprings are not identical to the parent, and we're going to see meiosis and mitosis play out in this. The advantage to sexual reproduction is that now we're going to create genetic variation. We're going to see that there's an enhanced in reproductive success in the environment. So we're going to see that due variation, species that have more variations are more likely to survive and reproduce, which will help them to evolve throughout time. So variation is very important for that. This advantage is located in a mate. So let's go over to meiosis. So meiosis, also referred to as meiotic cell division. Meiotic cell division occurs in the formation of gametes, sperm, and egg cells. Gametes are sperm and egg cells, I mean. These are produced for sexual reproduction. Some of the terms that I'm going to introduce or review in this section will be diploid, because in this process, a diploid cell contains a normal number of chromosomes. So in a human, what's the normal number of chromosomes? 46. So the cells that have 46 chromosomes are referred to as a diploid chromosome. But in meiosis, that diploid cell will then divide into 4 haploid cells, containing half the normal number of chromosomes. So this haploid cell forms in the gonads. This occurs when you're making the sperm and the egg cell. So this will occur in the testes, and if it's a female, in the ovaries. So meiosis takes place only in the gonads. Everywhere else in your body, it's mitosis. Right? From my head down to mitosis. No? Nobody get that joke? All right. Next time. All right, so all over your body, it's mitosis. Except for when it's time to produce the egg in a female, it's meiosis. When it's time to produce the sperm for a guy in the testes, it's meiosis. All the other times, I want you guys to think mitosis. So important things that you need to remember about meiosis. It helps to maintain a constant chromosome number at the time of sexual reproduction. And the fact that it increases genetic variation, which we'll see play out in evolution in our 3rd session. Let's go through the process of meiosis.
Meiosis is called reduction division because a single cell divides into 4 daughter cells. Each of the 4 daughter cells contains half the number of chromosomes that were present in the original cell. During new cell formation, the period of cell growth where the DNA is replicated is called intraphase. Intraphase is followed by meiosis 1 and meiosis 2. The first stage of meiosis 1 is prophase 1. In the prophase 1, the nucleolus disappears and the chromatin condenses to form chromosomes. During prophase 1, DNA is exchanged between homologous chromosomes. This is called crossing over. The centrosomes start to move towards the poles, organizing the spindle microtubules between them.
So those are centrioles again.
During prometaphase 1, the nuclear membrane breaks apart and disappears. As metaphase 1 continues, spindle microtubules attach to the centromeres of the chromosomes, and the homologous chromosomes line up along the metaphase plate.
Notice there's more than we saw in mitosis.
Metaphase 1 begins with the contraction of spindle microtubules, which separates the homologous chromosomes.
Homologous, you have two of the
The
same.
chromosomes start to move towards the poles. During telophase 1, the chromosomes reach the poles and the nuclear membrane reforms.
So in mitosis, it would stop here.
Cytokinesis 1 begins with the formation of
So you have
cell.
two cells.
As a result, two haploid cells are formed. Immediately, the two cells enter.
Oops. Oh. Oh.
During prophase 1, DNA is exchanged between homologous chromosomes, followed by meiosis 1 and meiosis 2. The first stage of meiosis 1 is prophase 1. Nucleus disappears, and the chromatin condenses to form During prophase 1, DNA is exchanged between homologous chromosomes. This is called crossing over.
Right. And I wanted to show you this. So this is an important part in creating diversity, is that crossing over process that's taking place. If you take a look, there's the pink represented by the pink shaded area. In that process, during the early phase of meiosis, we're going to have parts of the chromosome cross over to the other chromosome. This is one of the processes that creates diversity, create different characteristics in the cell. So crossing over leads to variation, and that's why meiosis is one of the key factors in creating genetic diversity.
The centrosomes start to move towards the poles, organizing the spindle microtubules between them. During prometaphase 1, the nuclear membrane breaks apart and disappears. As metaphase 1 continues, spindle microtubules attach to the centromeres of the chromosomes, and the homologous chromosomes line up along the metaphase plate. Anaphase 1 begins with the contraction of spindle microtubules, which separates the homologous chromosomes. The chromosomes start to move towards the poles. During telophase 1, the chromosomes reach the poles and the nuclear membrane reforms. Cytokinesis 1 begins with the formation of a cell plate.
All right, so what we saw before in mitosis, we end up with two cells that's identical. But in meiosis, the process is not going to stop here. There's two sets of division that's going to take place, or two series of division. So here, you have identical, but you're going to see that the process is going to take place again. So it's going to start at prophase 2, then metaphase, then anaphase, then telophase again. So it's two series of cell division that's going to take place.
As a result, two haploid cells are formed. Immediately, the two cells enter into meiosis 2. The four steps of meiosis 2 are prophase 2, metaphase 2, anaphase 2, and telophase 2, which are similar to mitosis. At the end of telophase 2, cytokinesis 2 takes place. As a result, four haploid cells are formed.
So that's how you end up with the haploid cell. If you saw at the end the chromosome, you had half the amount that you started with. So meiosis, a result of it, you end up with four cells having half the number of chromosome. This process only occurs in the testes to produce sperm, and only in the ovaries to produce eggs. So let's take a look at this. The process of creating the sperm or the egg is referred to as gametogenesis. But when it takes place in the female, it's referred to as oogenesis. And that's because you're going to produce one developed ova or ovum. And here, does anybody remember what these are called? Well, these are the polar bodies. And so these guys will not develop, they will disintegrate, whereas you'll have one developed ova. So here's one egg cell. For the guys, it's referred to as spermatogenesis, where all 4 of the cells mature into sperm cell. So to every 1 egg that's produced, there are 4 sperm that's made in spermatogenesis. And so we could see the process. Take a look at the chart on page 2. And let's talk about where they occur. So we're going to see once again that this is the egg production, 1 large, and here's the sperm production. Let's take a look at the process in more detail. Now at the top, the original cell. So let's say this is the testes, cell in the testes. It's referred to as a diploid cell. Another representation, and I want you guys to write this in, it's in your diagram B. That's going to be for spermatogenesis. You'll also see 2N representing a diploid cell. 2N means it has the full number of chromosome. N represents the number of chromosome. 2N means it's the full number of chromosome. Once it divides, it will divide into a half, which is represented with just the letter N. Sometimes you will see it referred to as a monoploid cell or haploid cell. So in spermatogenesis or gametogenesis, you go from a diploid cell into a haploid cell, from 2N to 1N. So in human, how many chromosomes would be in the haploid? 23. Because we said before, in a diploid there was 46, so it would be 2 times 23. Half of that, half of 46 will give you the 23. All right, and the same thing in the female. You have this diagram up top. You want to add in that the original cell is a diploid cell represented by 2N, and the bottom cells are referred to as monoploids or haploid cells. It's possible that there could be abnormalities in the egg or sperm cell. For example, in the case of Down syndrome, in the egg cell, you have the N, but there would be an extra chromosome. So we have N plus 1, and the sperm cell is still represented by N. So for Down syndrome, it will be 2N plus 1 in the zygote, in the developing cell. And so this is a picture of what the karyotype would look like. For Down syndrome, there's an extra chromosome on your 23rd pair of chromosome. Usually there's 2, mom, dad's information all throughout. Your 23rd determines what sex you'll be, if you're going to be a boy or a girl. But those who have Down syndrome, on the 23rd pair, there's an extra chromosome. Okay? So why don't we'll come back and pick up at this question, take a quick break. All right, you guys take 5 minutes, and we'll regroup. So we just completed our section on mitosis and meiosis. Quick overall review. Remember, the purpose is to not remember every single stage or phase, but to understand the purpose of mitosis, the purpose of meiosis, what's result of it, result of each, and how to apply each in the questions. So let's start off with a quick practice question to review what we've covered so far. So here, here's a diagram of meiosis in the ovary of an animal. It's shown below. In the answer space at the right, draw in the chromosome of cell A. Your drawing should show the usual result of the process of meiosis. So once again, here's the process of meiosis. Guys, just by looking at it, what type of meiosis what process is taking place here? What type? Right, so this is oogenesis. Right away, you should identify that there's 1 developed cell and 3 underdeveloped. So now the trick will be, let's talk about it first before you start drawing. What do we expect as a result of meiosis? What should be as a result of meiosis? What do you expect to see? If you forget, just go back to the previous page. What should we see as far as the chromosome numbers? All right, should be, and it doesn't 23 just relate to us, but there's some organism with a chromosome of 8, chromosome of 12. So just get an idea that it's going to have half the number of chromosomes. So if this represents the chromosome that's in the original cell, the parent cell, I want you to think of what would be the chromosomes in the resultant cell, the offspring, which would be the developing egg. Take a second, analyze it, think about what's supposed to happen. Look at what's included in the top drawing. And then go ahead, take a minute and draw it in. I gave you space to fill it in on page 2. If that circle is too small, redraw it on the side. Yep. What do you think it looks like? And so we're practicing, so take a chance. If you get it wrong, it's not a problem, but at least you'll know that it's wrong and you'll be able to correct it. All right, looks like everyone's ready. So as a result, what should we expect in the bottom in general? If this is a diploid cell, this would be a haploid cell. Full number of chromosome, half the number of chromosome. So if this is a full amount, we would expect half. So you would have one of these half shaded, and one of these fully shaded. So that should be your result, half of whatever's in the original cell. Good. How'd you guys do on that? Good? Okay, so we're going to take a look now. This is the second half of what we're going to do today. We're going to take a look through the different body systems and talk about the process and how all the systems are connected. So we're going to start out with the male reproductive system. And let's just take a look at the process that occurs.
Sperm are produced in the testes.
So
They are stored.
there's a space to write notes in.
During ejaculation, sperm are expelled from the epididymis and moved towards urethra through a tube called vas deferens. Along this path, the secretions from seminal vesicles and prostate gland mix with sperm and form a fluid called semen. Finally, the fluid is ejaculated through the end of the urethra.
So now you guys have the parts to label on the diagram. These are popular pictures to see. So let's take a look at it. A, what is A representing? A would be this area here. You guys remember what that is from class? Yes, this is the bladder. So you could go ahead and fill it in if you didn't remember it. B represent these tubes here. Anyone remember? No? The vas deferens. Vas deferens.
It's flat, right?
Yep.
Very flat.
Vas deferens, yep. Here we have C representing. Do you remember what C represents? Oh, that's the urethra. The urethra. We'll see that again in the excretory system. D representing the penis, and E is representing the testes. So quick notes to yourself. What's the function of E? Make note to yourself. What's the function of structure E? Yep. It's the site of sperm production. B, the purpose of B, this is where the sperm will travel up to where it will mix with seminal fluids from the seminal vessel, the prostate gland, creating semen. And the urethra is how it exits through from the body. So a lot of the questions that you'll see on the test coming up in a few weeks will be centered around the testes and the vas deferens. Let's take a look at the female reproductive system. Let's review the parts. And then
External structures of female reproductive system are mons pubis, clitoris, labia majora, and labia minora. The internal structures are uterus, fallopian tubes, and ovaries, which produce the female egg cells. The cervix connects vagina with uterus. Fallopian tubes lead from the ovaries to the uterus. At certain intervals, ovaries release egg that meets the sperm, and fertilization takes place in the oviduct.
all right, so let's review some of those sections. A, what does A point into? From your image, anyone remember what A is? Review from class. Look, A is the oviduct, also referred to as the fallopian tube. Important, there are a lot of questions on, or we do see questions often on the oviduct, because here you want to add in, this is the site of fertilization. This is where you see the sperm and the egg unite. So some of the most popular questions about the oviduct will be due to the fact that fertilization takes place there. B, what does B point into? Ovaries. And what's the function of the ovaries? Produce the eggs. C, the vagina, or also known as the birth canal, will be referenced. D represents the uterus. And so the uterus, you want to add in the definition as in, that's the site of embryo development. And so there's a couple of things that will happen. If you take a look inside of the ovaries, a female is born with pretty much all the eggs that she'll have for all her life. But it's the onset of puberty that will start the maturation of each egg. And so they go through different phases. The phase is dependent on the secretion of hormones such as LH and FSH. These hormones control the phase or the maturing of the ovary, where there's a follicle stimulating phase, and after the follicle stimulate, halfway through the cycle, which is usually day 14 of the cycle, the egg is released from the ovary, and that releasing process is referred to as ovulation. The egg is then released into the fallopian tube. After the egg is released, the area that it's released from is the corpus luteum, and that actually closes up. And so this is controlled by the menstrual cycle. The hormones are actually released from the ovary for the uterus. Does anybody remember the two hormones that's secreted from the ovaries? Estrogen is one, and the second one? It does. Progesterone. All right, so estrogen and progesterone, they're going to affect the uterus. So once again, LH and FSH is from in your brain. That's going to stimulate the production of the eggs or the maturation of the eggs. And then estrogen and progesterone is produced by the ovaries to get the uterus ready for the egg in case there's fertilization. All right, let's do a quick review question on this. Here, common images to see on the state exam. Here's the male reproductive, female reproductive system. Now, you have to think right away, as soon as you get the images, you should start labeling, figuring out what parts they're pointing to. Anytime there's a diagram, try to label it ahead of time. And then go down and look at the question. If there's a blockage in structure A and structure B, what would it most likely interfere with the ability to do? So take a second and think of that. So remember to try to label the sections and think about the function of that area first, and then how could it be affected. Yes? Which part? So C is the urethra, and what's the other one? B is the same thing as over here, the vas deferens. There's two on each side. That's the uterus. So what did you guys come up with? What if there was a blockage in this area? First of all, what is this area that they're pointing to? What is that to? Right, what's the function of the vas deferens? All right, let's go over to the female. What is this area that they're pointing to? The oviduct, fallopian tube, and the purpose of that site of fertilization, but also if there's no sperm to fertilize the egg, it's the passageway for the egg to get into the uterus. So if that area is blocked on both male, female, now I'm asking you to combine the two. What do you think would happen? All right, but still here, most directly, what's affected? Right? That is a possibility because that's after it's released from the body. So here, this blockage is going to prevent the transport of the gametes. This is going to prevent the transport of the sperm cell. This is going to prevent the transport of that egg to meet up with that sperm cell. So any blockage in both these areas is going to interfere with the ability to transfer or transport the gametes. Okay? So you're right by saying that it's going to interfere with fertilization, but we're talking for the guy, the sperm cell has to get out first. So it's going to block the movement. Let's see what we got here. So now let's talk about fertilization. So we have the sperm cell here represented with N, representing half the number of chromosome. The egg cell, half the number of chromosome. When the sperm cell and the egg cell fuse together, that's referred to as fertilization. When they combine, they form 2N. So the purpose of this is to restore the normal number of chromosome. And so the cell that it's restored in is referred to as the zygote. So the cell that contains all the genetic information needed for the offspring. So once again, dad's information, mom's information, combined in fertilization to create the zygote. The purpose, again, is to restore that chromosome number. Now, once it goes from fertilization takes place, here's your zygote. What's happening to the cell? Take a look at it. From here to here. You notice what's happening? From here to here. How many cells are here? How many here? More than 4. There's some in the background. Right? So around 8. So how did we get from 1 to 8? Let's review.
Mitosis.
Yes, good. All right. So mitosis is where the cell divides. You make an exact copy. So what's happening is each time the cells are going to get smaller. So you're going from that one cell dividing into multiple cells. So as the cell divide, it curves, it indents in to create more surface areas to make more cell. So at one point you guys were made up of one cell. How many cells do you have now? 2. You can't count. So you went from one cell to so many that you can't see it, you can't see them with your natural eye, and you can't count them. So that one cell divide over and over, and each time it divided, each cell got smaller and smaller. And then that gave way to different parts of the cell. So we're going to see that the cells on the outside will end up becoming the outer portion of your body. So here we go. These are going to be your ectoderm cells. These guys here will become your skin, the outer portion of your body. These guys here would become your internal organs. Those underneath here will become those like the tissues that cover those internal organs. So this process that's taking place here is cleavage. It's the rapid cell division that's going to help you to grow. So we're talking about you going from that one cell, which is a zygote, to many. So let's take a quick look into cleavage.
Fertilization. Zygote divides into tiny cellular units. This process is referred as cleavage. Zygote undergoes mitotic cell division to multiply. Here, the cytoplasmic growth is arrested. So, the cell size will not increase, but proliferate in their count. Cleavage process produces blastomere. Blastomere form blastula in the end and leads to gastrulation.
So here are the different layers. So you could see the different layers that's formed. So we went from that one cell to the point where we have so many cells that they're starting to form different layers. And so those different layers, the creation of different layers is referred to as differentiation. This is one of the popular topics to cover on the test. So it's important that you understand this concept. So differentiation is the process of transforming, developing cells into specialized cells, different structures with different function. So we're going to see that based on the location, the ones on the outside will become a specific type of cell. So here I have them, the ectoderm will be the external. So the external area of that ball will become the skin cell, the nerve cell, your muscle cell. I'm sorry, your skin cell, your nerve cell, pigment cells, those are all on the outside of that ball. The ones that are inside, in the middle layer is referred to as your mesoderm. So that's going to create your red blood cell, your skeletal, your muscle, smooth skeletal cells. Your ectoderm, it's the area in between that's sandwiched, has the mesoderm in the middle. So we have ectoderm on the outside, endoderm inner, and the mesoderm is lining in between the middle. So here, if it was a sandwich, you have the ecto and endoderm on the outside, and the mesoderm is the middle. And the ectoderm is where you get your lung cells. So you have some of your organs here, pancreas, your thyroid cells. So differentiation is where you go from that one cell into creating different types of specialized cell. This is called gene expression, where different parts of the genes are expressed. And remember, the gene is found on that DNA. So different parts of the DNA code for different genes. Based on the location of these cells, you create different types of cells. One of the most popular questions or topics to cover. So if we're looking from last review session 1 to now, this is how we're developing, this is how we're organizing the idea. We took a journey through the cell. The cells will come together and form tissue. Those cells will form tissue. The tissues will form the different organs in your body. Those organs will form organ systems, and all of those systems will work together to form the organism. So some of the cells that we'll see due to differentiation, the skin cell, nerve cell, muscle cell. Development will take place inside of the uterus. So we're going to see all these organs being formed inside of the uterus. So let's go back. Here's the diagram we have. Inside of the fallopian tube, we have fertilization taking place. Let's see if we could label some of the parts. What's going on at A? What is A pointing to?
Ovary.
Good. Ovary. B. Because it's being released from the ovary. It travels up here. Here the sperm cell. The sperm cell is attached to what? To the egg cell. What process is taking place in C? Right. After fertilization, that once they join, the egg and the sperm cell then forms. What is this cell now called? Before that. Before the embryo is formed. That's the zygote. That's the fertilized egg. It will move down into the uterus, get embedded because estrogen and progesterone has prepared the uterus for the implantation of the dividing cell. It will stay here and develop. So take a look. Here's the developing process. So let's see if we could label some of the parts here. Here, does everybody know what 1 is? So this whole thing here is the uterus. So then at the tip here, this is the cervix. Right? 2, here's the connection between the fetus and the mother's blood supply. This is the umbilical cord. I almost said it. What is 3 pointing to? What's that thing that the umbilical cord is connected to? Anyone know? That's the placenta. And so the placenta carries the nutrients, gases to the baby. That's the baby's lifeline. That's how the material is going to pass from the mom into the baby. That's how she's going to maintain it. And because of that, if the mom is consuming alcohol, drugs, smoking, any of those toxins that gets into her blood will pass through the placenta and diffuse into the fetus itself. And we could see where that could create malfunction. It can affect mitosis from occurring. It can affect how the chromosomes will divide. 5 is the embryonic sac, fill of the amniotic fluid, and that prevents shock. So as the mom is walking and moving around, that helps to cushion the baby. All right, let's do some review questions on this. See how much you guys are picking up here. So here, practice. Here, the reproductive organs, they're going to go through a process to create the sperm and the egg. They will go through a process to form the zygote, which will go through a process to create the embryo. Once again, whenever you get a diagram, try to label the parts before you even look at the questions. So now that we've gone through to see what we have, what's being asked or what's shown in the diagram, let's go over to the question. Identify each process. That's process 1, 2, and 3. Then state why process 2 is necessary for sexual reproduction. Then state one difference between process 1 and the cell that's produced in process 3. Go ahead. This is on page 4 in your review packet. Remember, as we're going through this, guys, you want to star topics that you might realize that you're not as strong in, or things that you might want to review some more. Put a star next to it, and you could use your review book. You've all been given review books in your class, Regents practice book. All right, you could practice questions from your teachers. You could use that to review any of the units that you find that you're not as strong in. All right, because this is an overview of all of them. So let's take a look. Identify the process. Process 1, what's that? Reproductive organs, you get the sperm and the egg. What process creates that? What makes the sperm and the egg? What do you think? Take a chance. If you're wrong, it's okay. All right, but we're looking for a process that would make the sperm. Meiosis. Right, so meiosis, same thing. You're going to make the sperm and the egg cell. Now let's take a look. What's happening to the sperm and the egg cell? It's combining to form the zygote. So this process is fertilization. Now here we're going from the zygote, that one cell, into the embryo. I could use a couple words here, but let's hear. Mitosis could be used here. What else? Since I'm making the embryo, that long word that I said is a popular, one of the favorites to see. Differentiation. So why don't you add that in if you didn't put that one down. So mitosis, yes, but you could also see differentiation for process 3. State why process 2 is necessary in sexual reproduction. So why is this process important? What does this do? The sperm and the egg combining together. What does that reinsure or guarantee? Remember, that's going to restore the full number of chromosome, because you have half, and fertilization will restore that full number. What's the difference between the cells produced in process 1 compared to the cells produced in process 3? Process 1, what did we say process 1 was? Meiosis. So then what do we know about the cells made from meiosis? What do we know about those cells? They have half the number of chromosome. Good. Whereas down here, process 3, what did we say it was? Mitosis. And what do we get from mitosis? What do we know about those cells? They have the full number of chromosome. All right, so these guys have half, they have full. So this is how you can apply mitosis and meiosis. So this is why I said it was important not to remember each phase, but to remember the overall idea or the purpose of mitosis and meiosis. Let's do another one. I have a nerve cell, skin cell, and muscle cell. Explain why they're genetically identical. If they're genetically identical, how is it that they have different structure, they look differently, and they have different functions? Same genetic information, yet they look different and have different functions. How's that possible? Apply it to yourself. In your body, you have the same genetic information in every cell. But you have skin cells that look different than your nerve cell, than your muscle cell. How is that possible? You want to give me a term? What's that term I told you to remember? That give you the different types of cells. Yeah, it's good. Differentiation. Right? So differentiation, what happens is different parts of your DNA is expressed. So remember, based on where they were located, different genes are expressed. So if you were in the ectoderm area, only certain part of your DNA is read and expressed. If you were in the mesoderm area of that ball, only certain part of the DNA. So gene expression is determined by location, and some environmental factors will determine how genes are expressed. And it's for the same reason where in the fall we'll see plants, we'll see that the leaves will go to different colors because the temperature is influencing what genes are being expressed. So we see different colors in the fall. Right? Your teacher probably spoke about the Himalayan rabbit, how the cold temperature changed the white fur into dark fur. That's because the temperature, the environment can influence how genes are expressed, how they show up. And so it's the same thing here. Even though they have the same genetic information, based on where they were located, different genes were expressed. And that's why these cells have different shape and carry out different function. Very important question, guys. So you want to review this one if you weren't sure about it before. Oh, we have another review. Practice question. So right away we have a diagram. So what do we notice about it? There's something to label. What is this thing here? What is A pointing to? That's the placenta. So right away you should think of labeling the diagram. Here's your placenta. Start thinking about what's the function of the placenta. It's going to transport nutrients, gases, get rid of waste for the baby. And so there's the fetus in the uterus. So it said identify the structure of the uterus where the exchange takes place between the mother and developing uterus, and that's A, and that's the placenta. So you could write that in. You have a picture of this on page 5. Identify one harmful substance that can pass through this structure and describe the negative effect it would have on the fetus. So take a second and write this one down. That's supposed to be number 2. Name a harmful substance and how it may affect the baby. Right, give me one harmful substance the mom could use. Alcohol. Anyone else? Tobacco, smoking, drugs, all of those. What's the negative effect it could have on the baby? All right, so we could see things like brain damage. We could see low birth weight, developmental problems. These are all things that can result from this. So these are popular short answer questions to see on the Regents. All right, so we have the cells. We've gone through the cells. We're taking a look at the tissue, structures, organs, and we're going to get into the system. So let's do a quick glance at the tissue.
An animal body consists of 4 types of tissues: epithelial, connective, muscular, and nervous tissue. Both internal and external structures of the body are surrounded by epithelial tissue. Protection and absorption are some of the functions of these tissues. Connective tissues bind organs together, form blood, and store fats. Muscle contraction and relaxation occur with the help of muscle tissues. Skeletal and cardiac muscle tissues have different characteristics, but perform similar functions in bones and in the heart. While the nervous tissues facilitate conduction and the integration of stimuli, they also protect and support nerve cells.
So I want you guys to get the journey that I'm taking you on. There's a lot of material, but if you keep it organized in your head, all right, try to think of it in a process. It's going to help you make connection between the information. So we went through the cell, the different parts of the cell. Know that cells come together to form the tissue. Those tissues function together to form organs, and that make up the different organ system. So we're going to take a look at a few of the organ system. And for that, let me give you guys another 5-minute break before we jump into the system. All right, so go ahead. You can use the restroom, grab something to drink, and we'll come back. All right, so nobody wanted to leave, huh? No bathroom breaks? Come on in. All right, guys, let's go through this last section for today, where we're going to journey through all the body systems. We've gone through the cell. You've seen the different types of tissue. You've seen how one cell transfer into different body parts, different body systems through differentiation. And now we're going to take a glance through each system. It's going to be a quick review of each unit. So what I've provided for you is underneath each section on the bottom of page 5, each system, I gave you an overview of what's happening in each system. Please feel free to add additional notes to yourself. I'm going to guide you through this part since there's so many parts. I'm using videos for each section. So our first section we're going to start with, our first system is the digestive system, which starts here in the mouth. From the mouth, food is then passed to the esophagus, to the stomach, from the stomach to the small intestine. It's in the small intestine where nutrients are absorbed into the blood. And then the large intestine is where water is absorbed into the blood. And waste products will leave the body through the rectum and the anus. So the important parts for us in this, the parts that show up the most on the state exam, is the enzyme action that takes place in the mouth. As far as we're going to see that starch is broken down there. In the stomach, we'll see that proteins are broken down there. In the small intestine, it's the final stage. Starch, proteins, lipids, and fats. Sorry, same thing. Lipids are broken down in this area. All right, so that's what you want to keep in mind of those 3 key areas. Let's take a look.
In the following medium, you will learn about the small intestine. The small intestine is 7.5 meters long, and it is extremely coiled. The pancreas and the liver secrete juices into the small intestine. The walls of the small intestine also secrete juices. The liver is a gland which is located in the upper portion of the abdomen on the right side. It is reddish-brown in color. The liver is the largest gland in the body. Bile is secreted by the liver. The gallbladder is a sac-like structure which stores the secreted bile. The bile is used in the digestion of fats. The pancreas is situated beneath the stomach. It is large and cream in color.
So we're going to see the liver and the pancreas come into play near the small intestine. They're secreting digestive juices into the small intestine to help that final breakdown. Remember last session we spoke about starch being broken down into, do you remember what starch is broken down into? All right, no, that's the energy. What starch is broken down into a building block? Simple sugar. So starch is broken down into simple sugar. Protein, if we break it down, breaks down into, from last session, it's broken down into amino acids. Your lipids are in fatty acids and glycerol. So here in your body, we're recalling, we're reviewing that information from the last time. In your body, the pancreas and the liver will add digestive enzymes to help finalize that process.
The juice secreted by the pancreas breaks down the complex carbohydrates, fats, and proteins into simpler components. The stomach partially digests the food and pushes it into the small intestine. The intestinal juices in the small intestine act on the food and complete the digestion. The carbohydrates are broken down to glucose. Triglycerides are digested to fatty acids and glycerol, and the proteins are broken down into amino acids. Thus, you have learned about the small intestine.
So most of the question you'll see, if they show up this year, will be based on that biochemistry. Breaking down the macromolecules into these micromolecules. Starch, glucose, and proteins are the 3 main ones. And for the digestive system, you need to remember where they're broken down. Once it leaves the small intestine, Once it leaves the small intestine, it's going to get absorbed, or it's going to diffuse into the blood. You remember the definition of diffusion from the last session? The molecules move in an area of high concentration to an area of low concentration. So if we have a lot of nutrients in the small intestine that's high concentration, and it's going to diffuse into the blood, which is an area of low concentration. All right, so check your notes, add any additional information you would like to add about the digestive system. Let's go to the next system, which is the respiratory system. So the respiratory system, you need to be familiar with what it looks like. So we have the nasal cavity, pharynx, larynx, trachea. So here's the trachea in the front. The trachea branches out into the bronchi, the tips of the bronchioles. At the very tip of the bronchial tubes are the alveoli, or the air sacs. And that's what makes up the lung, those air sacs. So we're going to see that blood's going to be flowing through the lung to pick up oxygen and to get rid of carbon dioxide. So when you inhale, you're going to take in that oxygen. That oxygen will then go down through the respiratory system, diffuse into the blood. When you exhale, you get rid of the carbon dioxide. Carbon dioxide is a waste product from all the different systems in your body. Carbon dioxide is one of the byproducts we see from respiration. And so when you breathe out, that's the carbon dioxide that's leaving your blood, diffusing into your lung. So when you breathe out, you're pushing that waste product out. So here, let's get a detailed look. Let's watch this other video on it.
Gas exchange takes place during respiration. The gas is exchanged between body and atmosphere through blood. The partial pressure of oxygen is greater in alveoli than blood. Hence, the oxygen diffuses into red blood cells from lungs. Partial pressure of carbon dioxide is less in lungs than blood. Therefore, carbon dioxide diffuses into lungs from red blood cells.
So guys, this is the key to this system. A lot of questions you'll see on this concept here. So you want to make sure you have a full understanding of it. The fact that diffusion is going to take place where here are the air sacs in the lung, and you're going to breathe in, take that oxygen. Due to diffusion, it's going to go from an area of high, because you just breathe it in, it's going to move from an area of high into the blood, an area of low. Now this blood will flow to the heart, and the heart is going to pump it out all over the body, to different areas of the body, so that you can use that oxygen. Do you guys know what that oxygen is going to be used for? You remember? You touched on it last time. It's going to be used for respiration, for cellular respiration. Right? And the same thing, that blood that's coming to your lung, the blood is going to pump it, the heart is going to pump it to the lung. It's going to be filled with carbon dioxide. So then it's going to diffuse from an area of high into an area of low. When you exhale, that carbon dioxide will leave.
Pulmonary veins carry the oxygenated red blood cells to heart. Eventually, the heart pumps blood to other parts of the body through arteries. Carbon dioxide is higher in body tissue than blood. The carbon dioxide starts diffusing into blood from body tissue. The lungs replenish the blood with oxygen through gaseous exchange and removes carbon dioxide.
So key point as we're leaving the respiratory system is to remember it's that connection with blood. If you notice all the systems, there's a connection back to the blood, and that's when we talk about making connection. How are all these systems connected to maintain homeostasis, to maintain balance in the body? Let's go over to the circulatory system. Here are some of the popular questions that you could see is based on what blood is made up of. What are the components? So there are three parts, three main components. You have your white blood cells. You have your red blood cells, which are usually round, disc shaped, smooth. Your white blood cells are usually irregular, so you want to add these in to your note. Platelets, these are the guys that help in the clotting of the blood. So if you have a cut, they're the ones that help to form the scab through enzyme action. And there's also plasma, that's the fluid that all the cells flow in. In the circulatory system, you also have the blood vessels. It was mentioned in the video before. The arteries, they're going to carry the blood away from the heart, whereas the veins, they're going to carry the blood towards the heart. So these guys that are bringing the blood back towards the heart are low in oxygen. So we refer to them as having deoxygenated blood. Right? And guys, your blood's not blue. Your blood, we're just looking at the flow that has less oxygen in it. Yeah?
It's representing oxygen-free blood.
Exactly. So it's that deoxygenated blood.
Why doesn't anyone want to know?
Huh?
Why doesn't anyone want to realize that the capillaries take the oxygen blood and convert it to blood that has no oxygen?
Deoxygenated. Right. But a lot of people think that their blood will be blue, but it's not. It's just representing, like you said, low oxygen. Whereas the artery will have oxygenated blood. It'll be full of oxygen. And so as we take a look at the blood, let's go back.
In the following media, you will learn about blood. The blood vessels consist of a constantly circulating fluid called blood. They provide nutrition to the body by transporting the digested food from the small intestine. Blood also supplies oxygen to various parts of the body from the lungs. Removal and transportation of wastes from various cells and organelles is also performed by the blood. Blood is filled with numerous cells of different types. The red blood cells are one of its types, which are composed of a red pigment called hemoglobin. The transportation of oxygen by the blood is accomplished successfully only when the hemoglobin binds with oxygen. The hemoglobin causes the blood to be red in color. Another type of cell is white blood cell. The white blood cells provide immunity by preventing the entry of pathogens into the body. When we fall down, blood starts to ooze from the cut. Later, a dark red clot appears, which stops the bleeding. The formation of a dark red clot is caused by another kind of fragment called platelets. The fluid presented in the blood is called plasma. The plasma holds the cells, which are suspended in blood. Thus, you have learned about blood. Red blood cells have the shape of a biconcave disc. The cell membrane has a bipolar lipid layer.
So I wanted you to see that it's a cell, it still has those cell membrane layers made up of the phosphate and the protein makeup. All right, so I want to review one of the state labs. The last time we reviewed diffusion through the membrane. Now, do you guys remember this state lab? Yeah. So this one of the labs that will be covered on section D or in section D on the state test. So you will not have to sit there and actually take your pulse rate at the exam. You'll be given a data table where you're going to have to interpret the information. So one of the things you guys did in class, once you found your pulse, you were given X amount of time, maybe 20 seconds, to count up how many pulse you found and you got in that time period. You were then asked to do it 3 times. then you had to figure out how many poles you would have in a minute. So you had to multiply it by 3 in the 20-second one in lab. You had to multiply it by 3, whereas here you would multiply it by 4, because you have to determine how many 15 seconds are in a minute, or how many 20 seconds are in a minute. So whatever the pulse is, so you could get a chart like this where the number is missing. And so this is why I said you need to bring a basic functioning calculator, so you could do the basic math just to make sure you have it right. I forgot that's not a smart board. I'm used to my smart board. Here, part B of that lab. After you took your pulse, remember you guys squeezed the clothespin? The purpose was to see the buildup of lactic acid, to see how long you could squeeze the clothespin, and to talk about what body systems were affected. You had to divide it into 2 groups. You had the group that decided to rest before squeezing the clothespin. Then you had the group that decided to exercise first before squeezing the clothespin. So the way you can see reflected on the test, you may receive a data table showing exercise and then after exercise, and you would have to compare to see which one was, which claim was right, if you exercise first or resting first. Another way you could see this information presented, here I like this one, where they show students that are athletic compared to students that aren't. So you've got to be open to seeing questions based on the activity, not necessarily the exact thing you did in class, but can you take that skill and now apply it to a concept or to a lab. It could be their height, it could be their age, but in this example it's whether you're athletic or not. Right, so this is a quick review of the Making Connections lab. Any questions on that? Right, so bring your 4-function calculator for just in case some of those questions show up. In the blood slide, it spoke about the white blood cell. One of the most popular units on the test is the immune system. The immune system, I try to break it down for you into 3 categories. The white blood cells, their job is to get rid of any foreign substance entering your body. So any pathogens such as bacteria, fungi, viruses, when they enter your body, they have different markers on their surface. That's different than yours. So those are referred to as antigens. So as soon as those enter your body will recognize that those foreign proteins, those proteins are foreign to your body. So they're going to send out white blood cells to destroy it. So there are 3 steps in this process. It's possible that your first line of defense could get rid of an invader or an antigen. So that's referred to as our non-specific response. This is where your skin can block it. Our skin exfoliate. We lose cells all the time, dead cells. So we could lose an antigen if it's on our skin. In that way, our skin also produces sweat and oil, which will have enzymes that can destroy or kill that antigen. Okay, so I'm making this very simple for you because sometimes it could get a little complicated. Say this line of defense doesn't work. You have 2 other lines of defense. The second line of defense is non-specific. Say something break through the skin, so you're able to pass through that first line of defense, or gets past your nasal passage, the mucus and your cilia in your nose. Say they get past that line of defense. The non-specific response is where phagocytes, these are type of white blood cells, they will come out and just engulf and destroy any substance. But in doing that, they destroy some of your cells. And so that's when you'll see that you'll respond with a fever, or you may have inflammation and swelling. Fever, what a fever does is increase the body temperature to try to kill that foreign substance. So having fever within a specific range is a good thing because it's your body way of destroying the pathogen. But we have to be careful when the fever gets too high, because then it can affect the proteins in your body and your enzyme. But a fever is one part of your body's response to invader. Same thing as swelling, inflammation. The third line of defense is if that doesn't work, we have a third line of defense where it's called specific immunity. This is where we have a cell-mediated response and a humoral response. The cell-mediated response is where T cells recognize this invader. These T cells, you already have them in your body. So while your body's trying to find, you were born with these, while your body's trying to find maybe that one T cell, this is when you may have the stuffy nose, you run into fever, all of these symptoms are going on. But once your body finds those T cells, they're going to make copies of themselves and they will destroy the pathogen. The humoral response is where the B cells are activated in your body. Their job is to produce antibodies. So you have two things going on at the same time as soon as your body recognizes this foreign substance. The T cells, they're going to destroy and kill. The B cells, they're going to make antibodies, will also destroy and kill. Your body in turn will make memory B cells and T cell just in case you get exposed to that virus again. It'll be ready to attack and kill it. Okay? So that's in three ways. You have passive immunity and active immunity. Passive immunity are those antibodies that's passed on to you from your mom. Another reason to say thank you to mom tonight, right? She gave you antibodies. And so that's going to help you to fight off foreign bodies or antigens. Then on active ways, if you've been exposed to the virus, like you got the chicken pox, so your body will then create the antibodies to fight it off. Or some of you guys will probably receive the shot for chicken pox. You went to the doctors and got it. So that's one way to boost your immune system. A vaccine is made of a weakened or dead form of that virus or that pathogen. And so what it will do is trigger your body, your B cells, to produce antibodies so that if you're ever exposed to it, you already have the antibodies to destroy it. So the antibodies are very specific. Antibodies have to match up with the antigen. The same way we saw enzyme and substrate are specific, antigens and antibodies are also specific. So antibodies will help to destroy the antigen, and that will call in also the white blood cells to engulf and take them down. All right, so let's walk through these together as we finish up. Identify one way the immune system fight pathogen. What does the immune system produce in order to get rid of pathogens? Which produces antibodies. So antibodies. So you have these questions you can fill in as you go. Identify the substance in a vaccine that stimulates your immune system. What is in a vaccine? All right, so or weaken part of the virus. Describe the response of the immune system. You will produce antibodies as a result. Identify one disease that damages the immune system. What is that virus that damages the immune system? Weakens it. It's the HIV virus that causes AIDS. Guys, there's a lot of AIDS questions on the test, and that's because it weakens the immune system so that it cannot fight off even the common viruses. Your next system is the nervous system. You see that nerves will send messages from one neuron to the next, and that will cause your muscle cells and your skin cells to respond. The next system I want to look at, which is also a regulatory system, are hormones. We've spoken about a few of them in the female reproductive and the male reproductive system, but you have hormones throughout the
chemical compounds and they work
body.
Coordination of body functions. Secretions of two dominant glands, the pituitary and hypothalamus, maintain the growth and chief functions of an organism. Thyroid glands secrete thyroxine that controls the fat and protein metabolism. Adrenaline is the secretion from the adrenal glands. It triggers various systems in body to deal with emergency situations. Pancreas releases insulin that maintains the sugar level in blood. Reproductive glands such as testes and ovaries secrete testosterone and estrogen that changes body structure during adolescent period.
Right, so some of the most common hormones that you'll see, these are also popular topics on the test. You'll see insulin, and oftentimes it's shown in a feedback mechanism where you'll see if the blood sugar is too high, the pancreas will secrete insulin to lower the blood sugar. If it's too low, then the pancreas will then secrete out sugar, the pancreas will secrete less insulin for it to then return to a normal sugar level. You'll also see different patterns showing different hormones in a feedback mechanism where one will get stimulated to turn on the process, one will be stimulated to shut off the process. The excretory system, the main structure in the excretory system will be the kidneys. The kidneys where the blood is filtered out. And so the kidney, we'll see, oops, I'm going too fast. The kidneys is where blood filtration will take place. So if we take a look, I want to take a look at this. All of these guys are connected back to the circulatory system. For example, the digestive system. What does the digestive system send to the blood? You guys remember what goes from? So all your nutrients are going to move into the bloodstream. And I gave you this chart towards the back of your packet. The excretory system is where the blood gets filtered out. The respiratory system, what do they put inside of the blood? Or take out of the blood? The respiratory.
Oxygen.
So this is going to be your gases. So we'll see gas exchange. The endocrine system, what do they put in the blood? Talk about them: estrogen, progesterone, LH, FSH, insulin. All of those are examples of? Right. So the endocrine system will secrete hormones into the bloodstream, and then the blood will carry it to the target cell. In the Making Connections lab, your musculatory system, the blood will take gas and nutrients to your muscles to help you produce energy, ATP. Or that pain that you were feeling when you're squeezing the clothespin, if there's not enough oxygen getting to your muscles, you produce lactic acid. Your immune system is where you see your white blood cells, and the white blood cells help to create an immune response. All right, so guys, we got through, there's some review questions in your packet, but we were able to get through the tissues today, body systems that make up the organism. In section 3, we're going to look at the genetics, biotechnology, evolution, and ecology. And that will wrap up our session. Okay? Thank you for coming out tonight.
Expert teachers and educators at Learn America have created an online platform that helps students better prepare and improve performance in standardized tests in math and science. Step 1. Our unique methodology involves taking pre-assessment tests. Students take pre-assessment tests to determine their skills, knowledge, and readiness required for taking a course. Students are provided a detailed progress report at the end of the assessment to determine their strengths and weaknesses, and areas to focus on in order to achieve higher success in the course. Students access the courseware through the Learning Management System, or LMS, that is available anytime, anywhere, either through a browser or through the apps for the Android or iOS platforms. Students will have direct access to instructors, view any posted course announcements, and view a calendar of upcoming course events such as homework due dates, assessments, and so on. They may also download a learning report detailing any of their course activity, such as assessment scores, anytime. Step 2. Personalized learning plan developed based on the student's test scores in the pre-assessment test. The LMS includes an ebook for each course that provides theoretical concepts broken down by chapters and topics. Each chapter includes diagrams, graphs, or animated videos to provide visual demonstrations of the theory. The LMS platform has several features to make the learning process easy. Students can highlight important information, take notes, use the online calculator, and unit converter. Our expert teaching staff has created an extensive question bank that provides self-assessment questions in the form of quizzes after every chapter. These tests provide students a quick assessment of what they have learned in each topic or chapter. Students get to view the results immediately and provide an instant view of what specifically needs to be revisited prior to moving to the next topic. Step 3. Adaptive assessments and formative updates, where students then experience a series of assessments. Formative assessments are configured to reinforce the essential concepts behind major topic areas and to prepare students for more rigorous assessments as they march towards exam readiness. The assessment results allow students a full 360-degree view of the courseware to reinforce the learning, and step-by-step detailed solutions to reinforce problem-solving abilities. Faculty will use the formative assessment results to further guide the students. The students are then put through a more challenging adaptive assessment process. Much like progressing through a video game's levels, students climb the competency ladder by mastering one concept at a time. The adaptive assessments culminate in a mock final exam covering the complete course. Step 4. Interactive moderated online forum. Students get an opportunity to participate in online discussions. Here, they can post questions and share information with other students taking the course, as well as faculty and teaching assistants. Step 5. Progress report and intense interactive review dialogues. We provide a detailed, personalized progress report to view course progress. Students may participate in faculty-led review dialogues. This is conducted either in person or over an online conference. Step 6. Ready review. Towards the end of the course, as their test approaches, students take a final summative assessment that details specifically what sort of outcome they can expect from their exam. With this assessment, students can dig deeper into what they need to know. Practice exams are available anytime and can be taken any number of times.